Hello students, I welcome you once again to your botany class, and today I would like to start with a new chapter, chapter 9 of your textbook, which is the enhancement or the improvement in fruit production. And so today I would like to start with the topic plant breeding of chapter 9, and which carries 4 marks. Okay. So India is traditionally an agricultural country. It's traditionally basically an agricultural country, and many of the population depends upon the agricultural products and produce. But traditional farming, it's not able to meet the demands of the increasing population. It's not able to meet the demand of the ever-increasing population, as well as to provide enough food for even the animals. So one of the major problem before the country since independence was the production of sufficient food, to provide sufficient food for consumption to the people. And you know that today the population of India stands at 1.3 billion, and it's ever increasing. So there is an ever increasing demand of food and food products. Okay, so you also know that as limited lands are fit for cultivation, and as such, since there are only few lands or limited lands or acres for cultivation, as such, techniques have been started to increase the yield per acre. So to increase the yield of the land. So techniques have been started, and through the plant breeding techniques, several varieties of high yielding varieties of crops and disease resistant varieties of crops have been developed. And this has led to an increase in the production of food, which meets the demand of food, making the country not only self-sufficient in the food, but also producing surplus of food. So this phase of tremendous increase in food production is known as the Green Revolution. It's a period of Green Revolution, and this took place in the 1960s. And credit goes to, you know, that Dr. M.S. Swaminathan, who was responsible for introducing Mexican wheat varieties, which was a huge success in India. Okay, so because of the Green Revolution, or because of this plant breeding techniques producing high yielding varieties of crops, particularly wheat and rice, and disease resistant varieties of wheat and rice, Green Revolution was a success. So what is plant breeding? What is plant breeding? Plant breeding is the technique of improvement of economically important food crops. It is the technique of improvement of economically important food crops or producing new varieties of crops. It's a technique of improvement of economically important food crops or producing new varieties of food crops which are better suited for cultivation, which have higher yield, which have more nutritional contents, which are resistant to diseases. So it's a technique of improvement of economically important food crops or producing new varieties of crops which are better suited for cultivation, which have which have higher nutritional content, which are better suited for cultivation, and which are disease resistant, and which gives even better yield. Okay, so what are the objectives of a plant breeder? What are the objectives of plant breeding? Or suppose that a person is to carry out this program to bring out a new variety of crop. So what are the objectives to produce a new variety of crop or to improve an existing variety of crop? So as I've said, from the definition of plant breeding, the objectives of plant breeding are to produce crops which are high yielding, produce crops which are high yielding or produce better yield, which are resistant to diseases, and which have better agronomic characteristics like plant height, then branching, etc. Then which are resistant to extreme environmental conditions, or which are resistant to salinity, drought, and which are resistant to various kinds of insects and pest infestation. So these are some of the objectives of plant breeding. And keeping with these objectives in mind, a plant breeder carries out the process of plant breeding. Okay, so with these objectives, what are the steps that have to be followed in producing a new variety of crop or improving an existing variety of crop? So let's see what the steps one by one in this. Okay, the first step includes collection of variability. Collection of variability. So this is the first step in plant breeding, and you know that genetic variability is the root of all breeding programs. Genetic variability is the root of all breeding programs, and differences among the individuals of a population or species for a given character constitute the variability of that trait. Differences among the individuals of a population or species for a given character constitutes the variability for that particular trait or character. And many of our present-day crops, are derived from the ancient domesticated varieties through plant breeding. So plant breeding was in practice since time immemorial, and so these differences that exist in the individuals of a population or species for a particular character or given character consider the variability for that trait. In many crops, pre-existing variability is available from the present. Pre-existing variability is present from the past and present varieties of crops and its wild relatives. Okay, and so the sum total of all the genes, the sum total of all the alleles of the genes of the crop present in cultivation of the crop in present cultivation as well as in the past constitutes the germplasm. The sum total of all the alleles of the gene present in the crops that are in cultivation and that are present in the past, that was there in the past, constitutes the germplasm. And collection of seeds or plants with all the diverse alleles of the genes in the crop plant constitutes the germplasm collection. Okay, so this is one of the first steps in plant breeding, collection of variability. So the next step, after the plants have been collected or after the diverse plants have been collected for a particular trait, the next step is evaluation and selection of parents. Okay, so in the second step, evaluation and selection of parents involves picking up of the plants that are better performing, that are better from among the total collection. So in the second step, plants that are better performing or that are better than the others, they are selected to be used as parents. And while the rest are rejected, the rest which are referred to as the inferior ones or inferior plants, they are rejected. So these selected plants, they are preferred by growing them under, favored by growing them under selected environmental conditions. Okay, so after collection of variability, the next is the selection of parents, better plants, better performing plants, which are selected from among the total collection. They are favored by growing them under suitable environmental conditions, while the rest of the plants are rejected. Okay, so the next step now is cross-hybridization among selected parents. So once these parents are selected in the second step, the next is to cross them, cross-hybridization among selected parents. So what is hybridization? Hybridization is the technique of introducing characters from desired parents into a single individual, which is also known as the hybrid or offspring. So hybridization involves the introduction of characters from desired plants or parents or species into one individual or into a single individual, which is also known as the offspring or hybrid, or this can be also carried out to bring out new genetic variety of crops. And as I said, the new genetic variety of crop or the improved variety of crop should be having better yield, should be resistant to diseases, should be able to adapt to diverse environmental conditions, or able to grow in diverse type of locations. Okay, so cross hybridization is the third step, and there are different types of cross hybridization. It is of different types: cross hybridization among selected parents, or crossing between parents, or between plants, or between species of different types. These are intravarietal. This intravarietal hybridization is the cross between individuals belonging to the same variety. Okay, cross between two individuals of the same variety is known as intravarietal hybridization. And another type of hybridization is intervarietal hybridization. So in this intervarietal hybridization, it is a cross between two individuals belonging to different varieties. A cross between two individuals belonging to two different varieties. Okay, and the next type of hybridization is interspecific hybridization. So in this type of hybridization, it is the cross between two individuals belonging to different species. So it's a cross between two different species is known as interspecific hybridization. And another type of hybridization is intergeneric hybridization. So cross between different species but belonging to the same genera. So the two plants involved in this type of hybridization, they belong to the same genera, but they are of different species. Different species, but same genera is known as intergeneric hybridization. So these are some of the different types of hybridization. Okay, so what are the steps to be followed during the process of this hybridization? What are the steps that a person or a plant breeder follows for a successful breeding of a new variety of crop? So the first is that selection of parents. The parents, that means the male and female parent are selected and they are isolated from the rest of the plant. So selection of parents, the male parent and the female parent are identified. The two plants to be used as male and female are identified and they are separated from the rest of the plants. And the next is emasculation. So emasculation is the removal of stamens, in a bisexual flower. So many of the plants that are used in a breeding program, they are bisexual. They are bisexual, that means that they have both male and female reproductive structures. And you know that in a bisexual flower or in plants, the stamens constitute the male reproductive structure and the carpel or the gynecium constitutes a female reproductive structure. So in order to cross between two desired plants, male and female parents, so in one flower, the plant that is, particularly in the flower that is to be used as a female plant or female parent, the stamens are removed. The stamens are removed so that self-pollination does not take place there. So that is known as emasculation, removal of stamens in a bisexual flower. While in many of the crop plants, the stamens are sterile. In many of the crop plants, the stamens are sterile, and as such, in a breeding program, those male sterile flowers, they are used as the female parent or the female plant. Okay, so after emasculation, the next is bagging. After the stamens are removed, the female flower is bagged. They are covered with polythene bags or they are covered with paper bags to prevent from external contamination or to prevent from pollination from undesired plants. So the female flower is covered with polythene bags or with paper bags to prevent external contaminants of pollination from undesired plants. Okay, next step is tagging. So this tagging involves putting tags on the bagged flowers, putting tags where it carries certain information as the date of emasculation, the date of bagging, or the steps that are being carried out, or the name of the female parent, or the name of the male parent. So this tagging involves putting tags on the bagged flowers and carrying information about this hybridization program. Okay, so and the final step is pollination. So once the pollen grains are matured and the stigma is ready, is receptive, or stigma is also matured, the pollen grains are collected from the male plant, the desired male plant, and the polythene bag or the paper bag is removed from the female plant, and the pollen grains are dusted on the stigma of the female flower. In that way, bringing about pollination. So in this cross hybridization, following the steps, only pollen grains from the desired male plant is allowed to pollinate the stigma of the desired female plant. So cross-pollination between two desired plants or two desired species to bring in desired characters into the offspring or hybrid of the hybrid. And the next step is, after cross hybridization is done, after the two plants, selected plants, after the male and female plants or the two selected plants are allowed to cross-hybridize, they are then allowed to undergo selection and testing of superior recombinants. So this step involves the selection of the superior hybrids. Once cross-hybridization is done, the offspring is produced, and so selection and testing of superior recombinants, the offsprings which are found to be superior, then the rest are selected and they are evaluated and selected. And this offspring or hybrid, they are found to have the characters of both the parents, while at the same time they are found to be superior to both the parents. Next, okay, even in these steps, the selected and they are also allowed to undergo selfing. They are allowed to undergo, the selected hybrids are allowed to undergo selfing so as to produce a uniform homozygous inbreed lines. The next is testing, release, and commercialization. So once the hybrids are selected, then the selected plants are tested in research fields for their for cultivation in different types of climatic conditions, then for the yield, the color, the seeds, then the disease-resistant character, or any other characters that the objectives of the plant breeder has. So they are tested in research field, they are grown in research fields for about 3 seasons in different localities, and then after the plant breeder has found that they are found to be better performing than the existing plants, they are released for commercialization. And so this release for commercialization is done by ICAR New Delhi, Indian Council of Agricultural Research. They carry out the process of evaluation and assigning name to the new variety of crop, and then subsequently they are released for commercialization or production for the farmers. So these are some of the steps that have been carried out, or they are carried out to produce new variety of crop or for improvement. So with these techniques, with these steps, disease-resistant varieties of crops have been developed. Then plants with better nutritional quality have also been developed, and plants that are resistant to insects and pest infestation have also been developed. And some of the varieties of crops that have been developed through these plant breeding programs are wheat, like this Kalyan Sonalika, then rice like IR8, then Jaya, Ratna, then different varieties of sugarcane has also been developed, and millets like Basra, and also new varieties of millets have also been developed through these breeding programs, because many of the crops are susceptible to different kinds of diseases. They are susceptible to insects and pest infestations. They do not have good protein quality. So through these breeding programs, these objectives have been achieved. And also there is another process known as biofortification. Biofortification. It is a breeding of crops with higher vitamins, mineral, and oil contents. It is a technique of breeding crops with higher vitamins, minerals, oil, and protein contents, which has now been able to meet the quality of food for the millions of people around the world. Because many of them, they are not able to meet the quality of food, but this technique has enabled the people to meet the quality of food. So in the end, plant breeding has made the country not only self-sufficient in food, but also has enabled the country, India, to produce surplus of food. Of course, there are still billions of people to get a quality food. So this biofortification has helped, and these are some of the steps: collection of variability, evaluation of species, evaluation and collection of parents, cross-hybridization, selection of parents, selection and testing of superior combinants, testing, release, and commercialization of plants are the steps to follow producing disease-resistant varieties of crops, and as well as high-yielding varieties of crops. So I went up this plant breeding program with this class here, and I hope to see you in the next class with plant tissue culture again.